Hi everyone, it's Mr. Vallejo. Welcome to class. Today in biology we'll be taking a look at cellular respiration. And so let's go ahead and go to our PowerPoint slides for today. So let's share that screen with you. Let me remind you, like I always do, that these PowerPoint slides are available for you in your learning management system, whether that is Canvas or in Schoology. So today's cellular respiration, including glycolysis, and going on to the Kreb cycle and the Electron Transport System. I think that in your particular course, this might be the toughest topic to comprehend and understand and learn. But I think that you have a good background now, especially since the last lecture when we took a look at some of the key background principles of energy and enzymes. Let's take a look at last time. What is energy? We saw that it's the ability to change and do work. We saw that there are a couple of different states. There's kinetic and potential energy, and there are many different forms of energy. We looked at the first and second law of thermodynamics. We said that the first law says that energy is not created, nor is it destroyed. And the second law says that things go from an ordered state to a disordered state. That second law is also called the law of entropy. Then we studied redox reactions. We saw how that transfer of electrons is moved from one chemical to another, from one molecule to another in a reduction oxidation cycle. And we saw the high energy electron acceptor NADH as a very good example of that we'll see today. We studied enzymes and activation energies, and we saw that enzymes lower the activation energy required to start a chemical reaction. That's a catalyst. And we saw that enzymes are organic, that they're proteins, that these proteins have active sites on them, and that the rate of the reaction is affected by many different things like temperature and pH and concentration of certain chemicals. Then we looked at ATP as well. So let me remind you about ATP specifically, because that is tantamount today to understand today's topic. How many ATPs can be made from one glucose? Well, turns out that we'll see that it's an awful lot. How cells make ATP? Well, cells make ATP through two different mechanisms. One is the substrate level phosphorylation, and one is chemoosmotic generation of ATP through a membrane. So first, let's look at substrate level phosphorylation. This represents this moon-shaped thing here. It represents an enzyme. And in this enzyme is this active site. And this chemical here, PEP, sits right in the active site of this enzyme. Now over here, here's another active site. This is where ADP sits. And we say that this is a coupled reaction because this reaction is happening right here, the phosphorylation of this phosphate. So this phosphate group is going to move, but it's going to move over to this chemical in this other reaction. So as this phosphate group moves over to here, that creates ATP. So we have two different chemical reactions. We have PEP to pyruvate, and then we have ADP to ATP. But these two reactions occur together, so we say they're coupled reactions. Now, through substrate level phosphorylation, what's going to happen is we're going to be able to generate 4 ATPs from the reactions that occur in what we call glycolysis. And also we're going to see that some ATP is generated in another set of reactions called the Kreb cycle. So this is going to result in 4 ATPs, 2 ATPs in each of those cycles I mentioned, and 4 total for one glucose molecule. Now, that doesn't sound like an awful lot, but what's going to happen is chemoosmotic generation of ATP is going to result in the rest. Now, we say it's chemoosmotic, and we know these terms already because we studied them in this course. We have a chemical gradient, and we have an osmotic gradient or slope or a change in between before and after. So you have a chemical and osmotic gradient. Over here, you have a lot of protons on the outside of this membrane. This membrane is the inner membrane of the mitochondria. Over here, on the inside of the inner membrane of the mitochondria, this area here, which is called the matrix, there's fewer protons. So these H pluses are going to want to go right through there, through this transmembrane protein, this transport protein, this ATP synthase, which is an enzyme. You know it's an enzyme because it ends in ASE, and what this enzyme is involved in is in synthesizing ATP, hence the name ATP synthetase, or the ATP synthase. Now, what happens is these protons move straight through there, and for each of these protons that moves through, one ATP is generated. That is to say, here, the movement of the proton in powers the phosphorylation of this inorganic phosphate so that it connects to this ADP molecule, and that creates the ATP, or adenosine triphosphate. Hence the phosphates here, you have 2 phosphate groups in orange, and here you have 3 phosphate groups in orange. But for each proton that goes through, you're going to get 1 ATP. Now, this diagram shows us the same location as the previous diagram. This right here is this right here. There's your ATP synthase again. Okay. Each NADH causes 3 protons to be pumped to the inner membrane. So if each NADH causes 3 protons to go to the inner membrane, how many ATPs are made from 1 NADH? Well, if 3 protons go through and there's 1 ATP per proton, that's going to result in 3 ATPs for each of the NADHs. Remember, the NADH is a high energy electron acceptor. But there's not just 1 NADH. There's going to be 10 NADHs in cellular respiration. So if we have 10 NADHs and we have 3 ATPs made for each NADH, guess what? That's going to be 30 ATPs. So that's much more efficient than the substrate level phosphorylation that we saw just a moment ago. So how many ATPs are made from cellular respiration? Well, 4 from substrate level phosphorylation. And then we're going to see that it's not just 30, but there's 34 from chemoosmotic generation, because there's another high energy electron acceptor that we're going to add to the story when we get to the Kreb cycle, called FADH2. So when you add those together, 4 and 34, you get 38. But we're also going to see that it's going to cost 2 ATPs to transport some of those NADHs from the cytoplasm to the mitochondria. When we're totally done with today, what's going to happen is I'm going to give you this diagram, or you're going to print out this diagram. And if you know this diagram, you will know cellular respiration as well, if not better, than any second-year biology, biochem student at a major university. If you can fill this in, this is what you need to know. So we're going to do this together in a little bit. But let me, in this talk, give you the nitty-gritty. Let me give you the information. Let me give you the details of the processes that happen in cellular respiration. We're going to take a look at glycolysis. Then after glycolysis, we're going to take a look at fermentation as a side trip. Here's glycolysis again, but after glycolysis, after you get the pyruvate here and here, there's 2 separate diagrams. We could go straight into cellular respiration, or could take a left turn and go into what we call fermentation. We're going to take a look at each of those options. Then we're going to spend more time on cellular respiration and go through the citric acid cycle, which is also called the Kreb cycle. And then we're going to take a look at how all those NADHs are going to get cashed in at the Electron Transport System. So that's where we're headed. I'm going to go and give you an overview of cellular respiration. Then we're going to go look at each of the steps of cellular respiration. We'll take a look at glycolysis in more detail. We'll take a look at fermentation. We'll take a look at this little part of the arrow right here. This is called the oxidation of pyruvate. We'll look at the citric acid cycle, or the Kreb cycle. and then we'll take a look at this area right here, which represents the electron transport system. Okay, so there are 4 different steps of cellular respiration, and we have multiple slides for each and every one of these steps, so let's take a look. During glycolysis, glycolysis happens in the cytoplasm and does not require any additional oxygen. Now, all the rest of the reactions happen in the mitochondria, and they do require oxygen. So when something requires oxygen, we say it's aerobic. So the oxidation of pyruvate happens in the mitochondria, and it's aerobic. The Kreb cycle happens in the mitochondria. It also requires oxygen. It's aerobic. And then the final step is the electron transport system. That happens in the mitochondria, and it's aerobic. So the last 3 happen in the mitochondria, and they require oxygen. The first step happens in the cytoplasm and does not require oxygen. So glycolysis, again, happens in the cytoplasm. It's independent of oxygen, does not require oxygen. So we say, here's a new term, it's anaerobic. So it's aerobic if it requires oxygen, just like if you were at the gym and you were working out and you were huffing and puffing during some kind of aerobic activity while you're breathing and you're requiring oxygen as you do that. This is the first step. This is glycolysis, step 1 of 4. In glycolysis, you're going to see that 2 ATPs are made directly and 2 NADHs are made. So that's glycolysis, and that's a scorecard for glycolysis. The next step is called the oxidation of pyruvate. It's just a little step. It's just one chemical reaction. It happens in the mitochondria, and it requires oxygen. What you see here in this chemical reaction, where you have pyruvate, and then you have the pyruvate converted to acetyl coenzyme A, what happens is you're going to lose a carbon dioxide, but you also are going to make some NADH. Remember, these are high energy electron acceptors, and for each one that you get, you're going to get 3 ATPs off of that eventually. Here's the Kreb cycle. The Kreb cycle, named after Dr. Krebs, but also I'll call the citric acid cycle because the first chemical here in the chemical reaction cycle is citrate or citric acid. And it's also called the TCA cycle because these chemicals right here are tricarboxylic acids. So that's what TCA stands for. So this happens in the mitochondria. It's aerobic. It's the 3rd step. Only 2 ATPs are made directly, but we're going to see that there's 6 NADHs that are made, and that's going to result in a lot of ATPs. Hopefully you can figure that out by now. You have 6 NADHs, and you have 3 ATPs per NADH. That's 18 NADH, 18 ATPs you can make on that. On top of that, you have a second electron acceptor called FADH2. You have 2 of these, and these are not quite as efficient as NADH. Instead of making 3 ATPs, FADH2 is going to make 2 ATPs. So 2 times 2 is 4. So if you recall the previous slides, a few back said that you're going to get 34 ATPs. Well, this is how you get your 34. You get 6 times 3, you get 18 there. You got 2 FADH2s give you another 4. And then from glycolysis and from the oxidation pyruvate, you've got 4 more NADHs coming in. And 4 times 3 is 12. So the electron transport system in the mitochondria is aerobic. It's the 4th step. And this is where actually 34 of the ATPs are made. You can see the proton pump. And what you see in this picture is that you have a chemical gradient. You have a chemical gradient and you have an osmotic gradient. And so these guys want to go back in to the matrix of the mitochondria, but they have to pass through this transmembrane protein, ATP synthase. So when that happens, you have the ATP is made when you have this proton passing through here, and then the ATP is generated. So let's take a look at these chemical reactions in more detail. First, you have glycolysis. Glycolysis, you have some reactions. Now, as a biology major, when I was sitting where you are, I had to memorize all these. And so you have glucose, goes to glucose 6-phosphate, goes to glucose 1,6-phosphate. Look, I'm not even not urine, it goes to fructose 1,6-diphosphate, P-gal, DHAP, and so on and so forth, and it keeps going and going. And so these are just things you memorize as a biology major, or as a chem major, or a biochemistry major. For our purposes, you need to know the end products. You don't need to know all of the reactions, and you don't need to know all the enzymes associated with the reaction. But the information I do want you to know is glycolysis means split lysis, we've seen that before, and glyco, that has to do with a carbohydrate, in this case glucose. So we're going to split this glucose. You can see glucose is a 6-carbon sugar here, and actually halfway down the chemical reaction it changes to a 5-carbon sugar, but then it splits up. So here you have 2 of these 3-carbon sugars, and so we split the glucose, and everything from this point on happens twice. So if you see right here, you can see that ATP is used. Well, that's not just one, but that's 2 ATPs used. And over here you have ATP is generated. So not just one, but 2 ATPs are generated. Okay. Actually, in this one right here, this is ATP being made. So you make 2 ATPs here, you make 2 ATPs here, but you lose 1, 2 there. So that's how you get your 2 ATPs all together. You're also going to have, remember, if you double from this point on, not just going to get 1 NADH, but you're going to get 2 of those high energy electron acceptors. So 2 ATPs are made, but ultimately 6 ATPs are made. And why is that? Because let's go back and look again. If we look at the yellow ATPs, here's lose 1, -1. Here's another one, lost, -2. Then you gain 2, which makes us even. And then you're up to right there. So 2 ATPs are made through substrate level phosphorylation. But then you have the NADHs. And the NADHs, remember, for each NADH, that results in 3 ATPs. So 2 times 3 is 6. Well, 6 plus these 2 is 8. But the trick is that these NADHs are in the wrong place. These NADHs, in order to cash these in, you got to take them to the mitochondria. Last week, someone called me up and said, "Hey, welcome. You've just won. Congratulations! A trip to the Bahamas. We have a cruise for you. That's 6 nights, 7 days from Fort Lauderdale to the Bahamas. Woo! I won a cruise. Isn't that great? Well, maybe not. Maybe not great, but at least a free trip. But hey, in order to get this though, all you have to do is get to Fort Lauderdale. Oh man, how am I gonna get there? That's gonna cost me hundreds of dollars. So yeah, it's not such thing as a free trip, but also with this NADH, hey, you're gonna get six ATPs off of those 2 NADHs. Nah, because you got to transfer. You got to take some energy and use that to move this from the cytoplasm to the inner membrane and the mitochondria. So it's going to cost you a little. It's going to cost you 2 ATPs, and that's why it's not 8, but it's 8 minus 2, which is 6 ATPs. So each NADH results in 3 more ATPs, but it costs 2 ATPs to transfer the NADHs to the mitochondria. All right, this is your scorecard. You need to know there's 2 ATPs, there's 2 NADHs, and that 1 glucose molecule gets cut into 2 3-carbon molecules called pyruvate by the time we're done. So that's what you need to know for glycolysis. Now, at the end of glycolysis, what could happen is that in bacteria and yeast, in the absence of oxygen, they'll go ahead and continue to process chemical reactions so that they can pick up more energy. Now, this says in bacteria and yeast that they'll form ethanol in a process called fermentation. Why do they even do this? Well, they do this so that they can pick up another NAD. Okay. Now, these NADHs are great. And remember, you get 3 ATPs for each of these NADHs. But if you can't get the NADHs to the inner membrane of the mitochondria, then it doesn't matter. It's like having a coupon for $100 off of, I don't know, 4 tires at the tire store. But if you don't buy any tires or you can't get to the store, you can't use the coupon. Same thing with these NADHs. You got these NADHs, you can't cash them in. Well, it doesn't really matter if it's a good deal and you get 3 ATPs off of this. Instead, these guys are waiting around and someone says, hey, instead of 3 ATPs, I'll change your NADH back to an NAD for you. And at first, that doesn't sound like such a good deal. But if you do change these NADHs back to NAD and pop them back in the second half of glycolysis, what happens is you have the chance to make another ATP. Another ATP is not 36 ATPs, but it's more than 0. So if you regenerate an NADH, so that another ATP can be produced from that NAD. Now, over here we call this chemical here acetaldehyde the final electron acceptor. These arrows represent the paths of electrons in these chemical reactions, and the electron finally stops here at acetaldehyde. So that's why we say acetaldehyde is the final electron acceptor in ethanol fermentation. Now, another type of fermentation happens in our bodies, in our muscles, and if there's a case where you can't get enough oxygen quick enough in your muscles, but you still need to go, like if you're running a really long marathon race or a half marathon or something, and after 50 minutes or so, your muscles are still going, but you totally need more energy. You've already maxed out the ATP stores that are in your blood system, and you need more. Well, what happens then is that we have what's called lactic acid fermentation or lactate fermentation. Same process that we saw on the previous slide, called fermentation. And for the same reason, you're going to take these NADHs, turn them back into NAD so you can regenerate an ATP. It used to be thought that lactic acid formation was associated with muscle fatigue. There are other reasons for muscle fatigue, but I know from practical experience that when I used to lift, the really huge guys would take a plate, like a 50-pound plate, and hold that straight in front of them so that the lactic acid would move out of their pectoralis majors, out of their chest muscles. So, wives' tale, well, it certainly might be one factor that has to do with muscle fatigue. All right, let's go to the second step. We have covered glycolysis. Now we take a look at the oxidation of pyruvate. The oxidation of pyruvate, once again, is oxygen dependent, so it's aerobic, and it happens in the mitochondria. It's the second step of the 4 steps that are part of cellular respiration. Some of your books, especially if you're using an older textbook, might call this decarboxylation of pyruvate or the decarboxylation of pyruvic acid. And we call it decarboxylation because you see that carbon dioxide going away right there. This does not produce any ATPs, but it does produce NADHs. It produces 2 of them. So from these 2 NADHs, you're eventually going to get 6 ATPs. So that's a pretty good deal. Why do we breathe out carbon dioxide? It's because of the carbon dioxide that comes out here in this step. And we're also going to see that some carbon dioxide is produced in the next step called the Kreb cycle. So this is actually a pretty short step, but some important stuff happens. Carbon dioxide and NADH2, we get 2 of those. Now here's the third step, the third set of chemical reactions in the Kreb cycle. In the mitochondria, we have the Kreb cycle, and this requires oxygen, and it's aerobic. And these are all the steps of the Kreb cycle, which is the third step in the cellular respiration cycles. So we're going to take a look at that in a little bit more detail. And so if you take a look at the reactions, you don't need to know the reaction, but if you ever do, if you dig this stuff so much that you become a graduate student in biology or biochemistry, this is the way I memorized it. Can I ask Sally for my ostrich? That's another one of those mnemonic phrases. So this is citrate, which is represented by the C in can. So this is citric, isocitric, alpha keto. This is succinic, fumaric, malic, and oxaloacetic. Oxaloacetic is over there. So this is a trick you can use to memorize just about anything. Make up a silly phrase, and then you can use that to help you remember things that you need to remember. Now, you don't need to remember all that. I might, but you don't. And so what you need to know is, well, what really happens during the Krebs cycle? What happens during the Krebs cycle is that you get 6 NADHs, which result in 6 times 3, 18 ATPs. You get 2 FADH2s, which is 2 times 2, which is 4 ATPs. And you get some carbon dioxide released as a byproduct. So there's more carbon dioxide coming out. That's significant, and that's why we breathe out carbon dioxide. On top of all that, you have 2 ATPs that are made through substrate level phosphorylation right there. So that's what you need to know about the citric acid cycle or the Krebs cycle. Now it's time to go to the bank. The bank is the Electron Transport System. This is when you take your Cuban money and you take it and you turn it in and you exchange it for Canadian money or whatever you're changing it into. So here's the Electron Transport System. You're going to take that NADH, you're going to take that FADH2, and you're going to exchange it for ATPs. These are the chemical reactions that happen. These are the reduction oxidation cycles that happen. These are the enzymes. You can see that many of them end in ASE. These enzymes that catalyze the reactions of the biochemical cascade that make up the electron transport system. Here's ATP synthase again. There's a nicer picture which shows you that transport protein. You can see these protons on the top that are moving to the bottom. And as they move from the top to the bottom like that, what happens is this guy spins, and as it spins, it produces an ATP by taking the energy from that proton movement in, uses that energy to phosphorylize and take that inorganic phosphate and adding it on to ADP to get ATP. Here's another picture showing that, and you can see that some of the enzymes that are involved are embedded in the inner membrane of the mitochondria. This one's stuck on top in an exterior protein in relation to the inner mitochondrial membrane. These guys are transport proteins within, and they are transferring those protons out. And finally, ATP synthase accepts those protons and is able to phosphorylize the inorganic phosphate, adds it onto ADP to make ATP. And just as we had a final electron acceptor in fermentation, we have a final electron acceptor in our cellular respiration cycle. That final electron acceptor is oxygen. As we follow all the arrows on the diagram, what happens is finally oxygen, right here in this diagram, oxygen is able to take those electrons that are traveling on these paths, and that oxygen combines with those electrons finally, and that makes water. Now, this water is unique. It's called metabolic water. You can see the chemical reaction right there. It is said that kangaroo rats don't have to drink because they'll make enough water this way. They'll make enough metabolic water to keep their bodies moving. That doesn't mean they won't drink. I did 2 weeks of desert research when I was working on my master's degree. One night and several nights actually throughout our stay, we had to trap kangaroo rats. And I can tell you that a kangaroo rat will drink water, but if it never does, it could still survive because of the production of metabolic water, and also by extracting water from their foods. Without making too much of it, what they do is they store their foods under the ground in their burrows, which have a higher relative humidity than the dry desert air. And so any moisture, even though it's a little bit of moisture, any moisture that's in the air in their burrows will actually go into their foods. And when they eat their foods, then they can extract that moisture right there. So overall, it says overview of glucose metabolism, the balance sheet, and you can see it adds up to 36. And if we take a look at it, you can see here's the 2 ATPs through glycolysis. There they are right there. Through glycolysis, you also make 2 NADHs, which result in 2 times 3, 6 ATPs. But remember, these NADHs are in the wrong place, so it's going to cost you 1 ATP each. That's why this is 4. In the second set of reactions, the oxidation pyruvate, no ATPs are made, but 2 NADHs are made. So 2 times 3 is 6. You get 6 ATPs. In the third set of chemical reactions, you got the Kreb cycle. And in the Kreb cycle, you got 2 ATPs, you got 6 NADHs, and 2 FADH2s. And then when you take all that to the electron transport system, you can cash in all these NADHs, all these FADH2s, and what you get then is over here, 6 times 3 is 18. Over here, you get 2 times 3 is 6. And then remember, FADH2 is not as efficient as NADH, so you're going to get 2 ATPs instead of 3 from an FADH2. When you add that all together, you get 36 ATPs. That's pretty tremendous compared to the 1 glucose molecule that you started with. Now, not everything you eat is carbohydrates. You also have proteins and fats in your diet. And you can take a look here. Here's your glucose molecule. And when you metabolize it, you get 36 ATPs off that. When you do that to a protein, you don't get as much energy. You get 12 ATPs. And when you break up a fat molecule, the fatty acids that make up a fat molecule are actually really good to store fat. So you can see here that 131 ATPs of that fat molecule. It's not as readily available though, and it takes a while, so that's why your body doesn't just do that all the time. Here's a diagram that says amino acids break down starting at pyruvate and acetyl coenzyme A. So they don't start at the very beginning of the reactions that we went over, but they jump in a little bit later, and you can see that in the next diagram after this one. You can see that fats also don't start from the very beginning, but they'll start at acetyl coenzyme A right here. And over here, you've got the glycerol and the 3 fatty acids. So this one does start pretty much back at the beginning, but not the whole molecule isn't. The whole molecule, when it breaks down, it's going to generate a lot of extra energy. It does it this way. So now that we've studied cellular respiration for glucose, we can better understand and more easily understand cellular respiration for different molecules like fats. And so you can see it again here. Here's your glycerol and your fatty acids jumping in at different parts of the chemical reactions that we just studied. So what we just studied is right here, glycolysis, there's pyruvic acid, there's acetyl coenzyme A, and the Kreb cycle, and the electron transport system. So that's what we just covered for a carbohydrate. But if you have an amino acid, you can see they jump in at different parts. If you have a fat, you can see the glycerol and the fatty acids are jumping in at different parts. All right, we do have a worksheet. It's called the Overview of Cellular Respiration that I'd like to do with you. We're going to review this worksheet together. And so let's go ahead and do that. If you have a copy of it, that would be great. If you don't have a copy, you might want to pause right here and get yourself a copy of that, print that out, or from your learning management system. All right, if you know this diagram, you know all the important things that you need to know in order to understand cellular respiration. So cellular respiration starts in the upper left-hand corner with the molecule glucose. I want to point out that this line here represents energy, and this line here represents a reaction as it starts over here, and as the reaction goes, we move along this line. Okay, so at the beginning of the chemical reaction, we have glucose. And glucose, in the first set of chemical reactions, which are known as glycolysis, you get 2 ATPs. And after you have your 2 ATPs, you also have the generation of a 3-carbon sugar called pyruvate. And not only do you have 2 ATPs, but you're going to get 2 NADHs off of that. And I want to remind you that you crossed the line right here. So crossing that line is going to result in an ATP being taken away. It's going to cost you an ATP to make that journey. So instead of making 6 ATPs from those NADHs, you're only going to get 4 ATPs. As we look at fermentation down here, we can see that there's different types of fermentation, and the different types of fermentation are the ethanol fermentation and the lactic acid fermentation. Both of these are anaerobic processes, and you're going to see that it does not require oxygen. So remember, we call those aerobic reactions because oxygen is not required. All the other steps 2 and 3 and 4, are going to require oxygen. So that's why we call those aerobic. And then remember, the first step happens in the cytoplasm, and all the rest of the steps happen in the mitochondria. So that's glycolysis. That pyruvate then goes on to the second step in the oxidation of pyruvate. I'll remind you that there's no ATPs that are made directly from this, but you do get 2 carbon dioxides and 2 NADH2s. So there's your NADH2. Remember, 2 times 3 is 6, so you're going to get 6 ATPs off of that. And you're also going to generate some carbon dioxide there. All right, in the next step, which is the third step out of 4, you got the Kreb cycle, the citric acid cycle, or also called the tricarboxylic acid cycle. You're going to get some ATPs. You have 2 ATPs that are made through substrate level phosphorylation. You've got 6 NADHs, and then you also have 2 FADH2s. So that is a lot. But those don't mean anything until you get to the last step. In the last step, you have the electron transport system, and you've got 10 total NADHs here. You've got 2 FADH2s right there. So you've got 10 times 3 is 30, and 2 times 2 is 4, which is going to give you 34 ATPs. So you got your 34 ATPs from the NADH and the FADH2 from the high energy electron acceptors. That oxygen that's right here is your high energy electron, it's your final electron acceptor. Remember, these arrows represent the journey of electrons as they're coming around and going through all these biochemical systems. And then finally, that electron ends up at oxygen. And when you add an electron to oxygen, you're going to get water, and that's metabolic water. All this put together is 36 ATPs from 1 glucose molecule. So that's very efficient, and a lot more efficient than just having substrate level phosphorylation that we saw at the very beginning. Alrighty, so that is our study of the electron transport system and the Krebs cycle and the oxidation of pyruvate and also glycolysis. And all that together is known as cellular respiration. I'm Mr. Vallejo. Thanks for listening. Hope you learned a lot.